everybody, this is Barbara, and this is the CIC study course, Certified Infection Control study course, and we're going to be talking about microbiology today. So in microbiology, we're going to be learning first of all about the bacterial cell. Now, all bacterial cells are prokaryotic. They have no nucleus, so they don't have a true nuclear membrane-bound collection of DNA and RNA like your cells and mine do. Instead, they have no true nucleus. They do have a cytoplasm, which is yellow in this picture. It's a gelatin-like matrix where the growth and metabolism and ATP production and replication does occur. So even though there's no true nucleus, there is a nucleoid in the bacterial cell. It's this collection of DNA and RNA. It's not membrane bound, but yes, bacterial cells have the DNA and the RNA, and it's localized in the nucleoid. Bacterial cells also have plasmids, which are these red circles. In this picture, they are subsets of DNA. Now, these are significant in the bacterial cell because they are often exchanged first in bacterial reproduction. So what this means is that if one of these plasmids contain the gene to make the bacteria resistant to a certain antibiotic, it's going to be exchanged first in bacterial reproduction, and this is going to lead to widespread antibiotic resistance in the population of bacteria that you are dealing with. Lastly, we have the cell membrane, which is this dark line surrounding the cell. A cell membrane is a bilayer of phospholipids and provides support for the cell. In addition to the other properties of the bacterial cell, we have the bacterial cell wall. The bacterial cell wall provides a barrier to protect the cell and is used to distinguish different types of bacteria. Primarily, we distinguish bacteria by dividing them into gram-stain, whether or not they are gram-positive or gram-negative. Gram-positive have a cell wall that has a thick layer of peptidoglycan, and it causes the bacterial cell to stain purple. Gram-negative bacterial cells have a thin layer of peptidoglycan, making them stain red. In addition, we have another differential stain which is used in microbiology to distinguish different bacteria and identify what kind of bacteria it is in a lab, and that is the acid-fast stain, which is used for bacteria with a waxy material in the cell, like Mycobacterium tuberculosis. So here's how the gram staining process works. So you start out with a slide, and you're going to put some of a culture onto that slide. And you're either going to have gram-positive, gram-negative bacteria on that slide. We don't know yet. So you're going to heat fix the culture onto the slide, and then you're going to add the first stain, which is the crystal violet stain, and it's going to cause both gram-positive and gram-negative bacteria to stain this dark purple. Then you're going to add the iodine, which is going to kind of lighten up the color, but drive it into the cell wall. Then you're going to decolorize, and the decolorizing step is where the big change occurs between the two different types, because you'll see there that the gram-positive are going to stay a light purple color, whereas the gram-negative are going to be indeed decolorized and going to be clear. Lastly, the gram-positive are going to stay that purplish color when the counter stain of red saffron is applied, where the gram-negative bacteria are going to take up that red stain, and gram-negative are going to be red. For oxygen requirements, we have the aerobic bacteria, which need oxygen to grow like we do, the obligate anaerobes, which do not require oxygen at all, the facultative anaerobes, which utilize oxygen if it's present, but they can still grow in the absence of the oxygen, and lastly, the microaerophilic, which only needs a small amount, hence the word micro. Spores are a dormant state, which do not have growth. So if a bacteria is in its spore state, that means it's not growing, and it's going to be resistant to environmental extremes and pressures, such as variances in temperature or moisture. Gram-positive bacteria are usually the ones that produce spores, like Clostridium difficile is a gram-positive bacteria, and we know that it produces spores, which makes it virulent. Now, this slide, so after you learn the material that's been presented in the previous slide, I want you to go ahead and look into your book, your ebook 6th edition certification study guide. And after you learn this material, then you go to this slide and you test your knowledge. So if you open up your book and you go to chapter 3, and you look at question number 14. So question number 14 in chapter 3 says, a microbe that can grow in the absence of oxygen but is also able to utilize oxygen for growth is A/An, A, aerobe, B, obligate anaerobe, C, facultative anaerobe, or D, microaerophilic aerobe. So we did just talk about this in the previous slide. So if you look to the previous slide, you'll see that it is C, facultative anaerobe. So the idea is that you learn the material and then you test your knowledge in the questions part of the last set. Good luck in studying, everybody.